Good morning. Well, here in Costa Rica, it will be good morning, good afternoon, everyone. I just want to double check. Are you seeing my screen this moment
Yes, we can
to
see us.
confirm? Okay, great. Thanks a lot. Okay, I'm going to start the first interviewer here. It's pleasure and an honor to be here and to share some of our knowledge about Brucella. That's the organism we work with. Just to start to talk about that, I want to tell you that Brucella is a gram-negative bacterium that infects mainly mammals and in them causes reproductive failure, abortion, and a reduction in the production. And that's very important because it causes a lot of economic losses. And something very interesting of Brucella genus is that it has several species and the species share around the 96 to 97% of genomic identity. But in spite of that, the biological behavior of Brucella species is very clear, very particular, with high host specificity and different pathogenic risk and aggressiveness when we talk about the development of the disease. So we have just 4 or 3% genome length to analyze in order to find some genomic traits that could help us to try to understand, explain a bit about that host specificity. And that's why we call it to find the needle in the hay, because it's very challenging to do that from the genomic point of view, because usually you look for similarities when you are performing genomic analysis. But in our case, we are actually looking for differences. And that's, again, very challenging. And that's why we call it the needle in the hay. And that's why my talk is titled Brucella Variability and Host Specificity, the needle in the hay. Today, there are 6 well-recognized species, Brucella species, by the International Taxonomy Committee of Brucellosis. These species are marked here with the blue dots next to the name of the species. And in the draw, you can see that there's an animal, a host species next to the name of the Brucella species. And the arrow here indicates the zoonotic risk, the risk of transmission from animals to humans, and the intensity of the color and the size of the arrow indicates how big is that risk for each one of the species. For example, Brucella melitensis infects preferentially goats and sheep, and is very pathogenic and very zoonotic. And that's a general description for several of the recognized species, and some of these species are still being proposed and not officially recognized yet. But you can see that the genus is expanding. That's why I said that infects mainly mammals, but there are other host genuses also being being included species that are affected also by brucellosis. Our studies focuses on 3 different species that are present here in Costa Rica, that are Brucella seti, that infects preferentially marine mammals, most specifically strip dolphins, Brucella abortus, that infects cattle preferentially, Brucella canis, that infects dogs preferentially. And in them, as I was saying since the very beginning, this infection causes abortion. And that's how the transmission occurs, when usually other animals or humans get in contact with the aborted fetus or membranes or fluids containing the bacterium. And that's how this is an occupational disease, almost always affecting people that work with animals or farmers. During mating, the animals can transfer the bacterium to each other, and this bacterium also sheds on milk. And that's how the consumption of unpasteurized milk production, milk products, excuse me, is the main cause of infection of people. And this disease is not like little, it won't kill people immediately. It develops as a chronic debilitating disease, and it forms part of the puberty cycle. And that's very important, and brucellosis is considered a neglected disease because it almost always affects poor people. So there are no much interest in working with that, as you can imagine, in the sense of directing funds to research and control this disease. And I was saying, here in Costa Rica, we have those 3 species that we have been focusing in. And I'm going to show you here a picture that is interesting for us in our environment, because it somehow supports a possible hypothesis of how Brucella can jump from one host to another. This is one of our Costa Rican beaches, and you can see that stranded dolphin here. People are curious, so you can see tourists here around the dock. And if you zoom in, you can see that there's blood around. These people can get infected. Actually, this dolphin was positive for Brucella sepsis. The dog, we don't know, but they are very curious. It's very likely that they will leak this blood. And if you see at the distance, you can see a group of cattle coming in. And actually, one is drinking water, some water that could be also contaminated with the fluids. And well, I was saying that this is a hypothesis, but it's a very interesting picture for us, because here it depicts the 3 likely Brucella species that are present in our country: Brucella bordeaux, maybe Brucella canis, and also Brucella seri. And that makes us ask how all this could may have influenced in the jump from one host to another. And I was saying that these likely species share a very high identity, very high genomic identity. And yet the specificity or preference of the host that's infected is very clear from the biological behavior point of view. And that very small amount has been explored and analyzed by different tools trying to explain this behavior. And several tools have been used: proteomics, pulse field, gel electrophoresis, Brucella, the restriction pharmacy, polymorphism analysis. Also the analysis of insertion sequences like IS711 that seems to be a specific transposon or insertion sequence that's specific for Brucella, and genome-based techniques. That's what we have used the most. Some of these include multi-locus sequence typing and multiple locus in a variable number of tandem repeat analysis, that's MELBA. And we have supported our analysis in these tools, but also we have done or performed a lot of whole genome analysis. And then we have performed the phylogenetic analysis of our samples in the world context. But first, I want to tell you that Brucella belongs to the alpha proteobacteria family. In this group, you can see that Brucella shares similarities or some familiar characteristics with soil and plant bacterium that have a very big genome, like Agrobacterium or Mesorhizobium, but also is close to bacteria that have a very small genome, like Bartonella and Rickettsia, organisms that are intracellular exclusive bacteria. And Brucella has an intermediate-sized genome conformed by 2 chromosomes that all together codify for around 3,500 genes. And has, well, 2 chromosomes, a big one, and a second one that seems to be megaplasmids. That's what's understood from the phylogenetic analysis. And I was saying since the very beginning, the species of Marcela, Marcela bortus, Marcela metensis, Marcela penis, Marcela seti share a 96 to 97% identity with a high GC content, going from 55 to 58% of GC content. What means that the genome is very stable. And that's why we have to look into that 3% for differences that could help us understand that very particular biological behavior inside of being so closely related and almost identical from the genomic point of view. And that's why we say that we are looking for the needle in the hay. With our studies, we have aimed to identify the elements that induce genomic variability in the Brazil genus. As I was saying, in order to understand a bit more about that specificity of our host preference and variation of pathogenic behavior. So we have, oops, excuse me, I had a problem with PowerPoint, as you can see. We have performed several analyses based on whole genome sequences analysis, and that's what I want. Excuse me, I hope it doesn't cause a delay. Everybody is seeing the screen again, just to check.
Yes, we can see yours.
Okay, yeah, thank you. Excuse me for my PowerPoint to fall down. So we analyzed our Costa Rican isolates and the whole worldwide context by analyzing and performing all the phenotypic characterization and also sequencing our isolates, and used the genomes that were available in databases, in public databases, like and the NCBI or DDI example. So we think that we analyzed some of them in a not very poor data set. So at the very end, analyzed 121 avoided genomes, 95 became, and 40 Brucella seti that are very rare yet in the databases. And we developed a workflow for this analysis. Once we had the reads, the genomes already sequenced, we align them to a reference and look for single nucleotide polymorphisms. That's something that's a little bit more of general knowledge. And we use that information to perform a phylogenetic reconstruction that allowed us to group and to compare the isolates based on similarities and geographic origin. We also aligned and assembled the genomes again by using reference and also the novel. We annotated the genomes and used that information to perform also a core genome analysis that allowed us to confirm the SNP phylogeny that we had produced. But also we looked for pseudogenes. We look for the presence or absence of genomic islands or shared anomalous regions. Not only the presence, but also the orientation and position along the genome. And we look for the number and position of that insertion sequence that I was talking about before. That's the insertion sequence 711. And we perform this for the three, individually for the three species, Brucella seti, Apolis, and Kimis. We looked for these. I'm going to tell you why and explain a little bit more of each one of them following here. We looked for the pseudogenes because it provides information of evolution. The pseudogene definition is a bit challenging also, and you can find different definitions in the literature. So this is what we used. It's kind of a fossil record of a gene that was previously present. And when you compare the gene that's in your organism against another one, you can see that definitely it's not the same gene that was originally. So there are several causes. One is a premature stop codon that induces the end of the gene and earlier in comparison with the original one. So you end up with a shorter product. It doesn't mean that it's not functional, but definitely it's not the same one that you saw in the reference genome that you are using to compare. You can see the absence of a stop codon. Same effect, you have a longer or a shorter coding region. You can have the loss of the promoter or the regulatory binding site that's earlier in the genome in comparison to that gene specifically, and finding these is particularly challenging. You can have functional domain changes due to SNPs. You can have frame shifts or frame shift plus partial deletions, but also insertions or frame shift insertions. All of these have also been proved to behave as microRNAs and then functional, but definitely it's not the same behavior that we had when we compare them with the original sequence. And something very, very interesting about developing the pseudogenes is that they have been associated with the genome reduction and host dependence development for the pathogens. You have already seen this phylogenetic reconstruction of alpha proteobacteria. I want to add here that mitochondria is considered an alpha proteobacteria, and it has a very, very small genome as it became an endosymbiont and an intracellular obligatory organism. And there is that relationship where the number of pseudogenes is related directly to the host dependence. So the higher the number of pseudogenes, the higher the host dependence, and the higher the possibility of becoming an obligate symbiont. So you can see here that organisms like Rickettsia or Bartonella that have a very small genome become obligate symbiont, while free-living bacterium like Bradyrhizodium or Mesorhizodium have a bigger genome. And this genome reduction and development or pseudogenes is associated with the presence of insertion sequences like the IS711. And that's why we also look for that IS711 pattern. The genomic islands are bordered also in the extremes with this IS711 that helps to introduce these segments of DNA along the genome. There are several genomic islands and shared anomalous regions that have been reported in Brucella, and its presence seems to be species specific. For example, here you can see this one that's named INC-P is absent in aborted melitensis, but is present in canines and microti, just to give an example. And this set of genes encoded into the genomic islands provide some properties to the bacterium. And the IS711 not only can help us to induce the development of the pseudogenes and the insertion of these genomic islands, but the number seems to be specific for the Brucella species. And we were not only interested in confirming the copy number, but also the position, because that would definitely modify the way the transcription is performed. So the main results of our analysis confirm the clustering of Brucella seti in this case, according to host preference and geographic origin. So in that very small amount of difference that comes from the 4 or 3% difference, you can see that Brucella seti from Pacific, and also most of them coming from Stenella alba, that the striped dolphin are clustering together. the same that we see from isolates from the Mediterranean and the Atlantic. When we look at the pseudogenes pattern, it confirms also the distribution seen with the phylogenetic analysis based on SNPs, and the pseudogenes are shared in branching points. What I mean here is that all of these isolates share the same pseudogene, and not only the same pseudogene, but also the pseudogenization cost. What tells us that it was not a stochastic event, but it was something that happened in the common ancestor and was fixated in the population. And in here we have 64 shares. I know this figure is a bit heavy. Here you can see, for example, the Brucella series representative of the Pacific plate, this P1. And here we have a color code for the function of the gene. So for example, here, this gene that has a function associated with wall membrane and motility is a pseudogene, and the cost is the same for all of the isolates. It was on frame shift. And that's the description for the pseudogenes that we were finding in the analysis. The IS 711 also shows a specific pattern that looks like a barcode with different or a specific number of copies for each one of the isolates, but also with some particular distribution. You can see here that the clades correspond with the presence or absence. For example, here we have these insertion elements represented by this peak in the very beginning of the genome that's represented concatenated in the top. And you can see this insertion element to be absent in the Pacific plate. And so for the other positions, we found also differences in the genomic island distribution. For example, here we have a deletion and we have an inversion here that's very important, and in the Pacific plate that is not present in the Mediterranean or the Atlantic. These are the genomic islands or concatenated. And I know this is a lot of information altogether. We have this already published in this paper that's available in 2017. And the other Brucella species confirm somehow this variability with the Brucella avarus phylogeny, where we found 5 different clades in our Costa Rican samples, a big one, but also some other smaller clades that cluster together with Europe and the US that are not weird for us because our cattle came from there. And we have the dispersion of these lineages all along the territory. That our territory is very, very small. We have this kind of supportive results also with the genomic island distribution, and similar with the IS 711 analysis. And the small amount of difference that enough final to perform a filotemporal analysis to define an approximate date of introduction of the clades in Costa Rica. So this is the main one. These are the clusters that come with European introductions. And something very interesting here is that our filotemporal analysis coincides with the historic records, because the reports of bovine brucellosis in Costa Rica was given for the first time in 1899, and that's the median estimation of our beast-based analysis, the Bayesian analysis, and that's something very, again, interesting for us because we have a very small amount of difference to perform this kind of analysis, but it provided enough information to perform a filotemporal analysis. Again, this is all detailed in our paper. And something similar happened with Brucella canis. We had a shorter or smaller number of samples, and we found three lineages that confirm the origin of our Brucella canis coming from the US. That's something that we were expecting, but we also found some maybe illegal introductions of um, animals from Mexico and Sweden because the clades were coming together. And this is published also. And some molecular characterization of Brucellavis in Argentina allowed us to perform also some distribution of the information. So the result summary that we have had so far is there's higher diversity among marine mammal isolates. That's something that we definitely were expecting because the host has not been manipulated by humans. That's something very different from cattle and dogs. The divergence that we are seeing is according to geographic origin, so that allows us to track the epidemiological origin of infections and control or call the attention and entrance points for the country. The clade dispersion is related to new introduction into territories. It also works as a gap and biosafety control. So the main conclusion is that our results establish basis for future analysis of Brucella genome because we use this workflow of SNPs to the genomic island and insertion sequence analysis that I was showing to you. There is more variability among Brucella 30 than among Brucella borders and Brucella canis. Brucella saying the hosts for Brucella 30 are wild animals. So there is no a bias in the immune system for these animals. And the variability is given by the pattern and number of SNPs, IS711 distribution and number, because it's not only important the number, but also where it is located along the genome, because it can modify the expression or the development of the gene, and by the genomic island and shared anomalous regions order. And definitely the variability is associated with geographic distribution patterns and host preference range, as I was showing to you in the phylogenetic tree. All of this is summarized in this review that was released last year. And just to close, I need to acknowledge all of the people that have helped an institution that has been involved in our analysis. So sampling the animals, performing pathogenic analysis, helping with training and providing the closer for the informatics analysis. And partly to show you the group, the working group, that's our group based in Costa Rica. This is Edgardo Moreno. That's a very heavy name in Brucellosis. He's already retired, but he still works just because he loves it. And now the head is Caterina Guzman. If you have any doubts, of course, I'm open to answer any of your questions if that's in my possibilities. And here you have my email and my research case profile if you have any doubts or want to contact me. Thanks a lot for the invitation, Joe.